Hello everyone, and welcome to today's live broadcast, Strain-Level Microbiome Profiling using rRNA Gene Amplicons, presented by Dr. Mark Driscoll, Chief Scientific Officer, Co-founder, Shoreline Biome. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen. Type your questions into the drop-down box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Mark Driscoll. I will now turn the presentation over to him.
Thank you, Kaylee. I really appreciate the introduction. I'd like to begin today by thanking our collaborators and the people who actually have done the work that I'm going to talk about today. Our collaborators at the Weinstock Lab, at the Jackson Labs for Genomic Medicine, Bo Yang, Yan Zhao, and Jethro did a lot of the analysis, and Dan Phillips did the heavy lifting at the bench. We've collaborated with Peter Setlow. You'll see some of George and Barbara's work here on opening cells and cracking microbes and getting the DNA out. I would also like to thank Dan Fasulo at Pattern Genomics, who has written a lot of the software that's doing the analysis that I'll be talking about. And at Shoreline Biome, I'd like to thank Chris and Ryan for all their hard work. So with that, the microbiome. Most people in the audience will be familiar with the microbiome, but just to remind people and get off on the right foot, I'd like to talk about why it's interesting. Humans are, in fact, a composite organism. You have 10 times as many microbial cells on and in your body as you do human cells. And it's extraordinarily interesting to think about that there's about 100 times the genetics going on inside those microbes as there is in humans. So if you want to find out about human health, it's going to help study the microbes. And in fact, a lot of the work that's been coming out in the past few years has shown that microbes, we always knew they influence digestion and health that way, but really regulating the immune system, participating in the training of the immune system during development, and resistance to pathogens, and they inhabit all the spaces on your body and protect you from invasion by unwanted organisms, and do everything from modulate your bone mass density to mental health. So really, microbes are extraordinarily important in overall health and disease, and people want to study them. But how do you generate a microbial profile? How do you figure out which microbes are there and what are they doing? So really, microbial identification is the key. So this is what we want to do. We want to find out who's there and what they're doing. And I'll be talking today about using the ribosomal RNA genes as a microbial mugshot. Essentially, I call it that because what you're doing is you're taking a look at a small portion of DNA and you're attempting to identify the organism that came from. So, how do people do this? What you'd want to do is target a gene that every microbe has. And in fact, the ribosomal RNA genes are in all of the bacteria that are out there. And so we target this gene because we know everybody's got it. So the gene itself, in purple here, are the parts of the gene that are invariant, that you can generate PCR targets to. And there are also 9 variable regions inside there, which when you flank these variable regions with PCR primers, you can read through with a sequencer and then determine the sequence inside that variable region and pin down which organism that belongs to. That's basically the upshot. Now, the more information you have, the better your accuracy is going to be. And part of the problem is that closely related organisms often has very similar 16S sequences. So the more closely related organisms can be difficult to tell apart. I'm going to discuss this in a little bit. But in the next slide, what I want to show is that this gene itself is interesting in that it has a 2-dimensional and 3-dimensional structure. And the ribosomes are interesting, right? Because the ribosomal transcription product is actually directly forms the molecular machine that makes proteins. And so this gene product will form a 2-dimensional folded structure shown on the left, and then that is base paired. And the primer sequences that we target the invariant regions are, you can see those primer sequences will also reflect that base paired 2-dimensional structure. And that in turn folds into, on the right, you can see the 3-dimensional structure of the final 16S ribosomal RNA. But the 2-dimensional structure is important to think about because that's vital to the functioning of the ribosome itself. And that's where our primer sequences are going to be targeting. A little more on that in a bit. But for microbial identification, since these similar organisms can have similar 16S genes, you can see in the case here, I've shown 4 different Clostridia, and they cause actually 4 different kinds of disease. And so it would be very useful to know when you're trying to identify organisms, not just that it's a Clostridium, but that it's a Clostridium difficile. And in fact, we can do this with 16S and 23S genes. But you require a unique, rather unique amplicon design that I'm going to discuss in a bit. But not only is it useful to know the genus and the species of something like Clostridium difficile, there are different strains that are involved, and some of these strains can carry toxins and some of them don't. So even if you have the species, it could be interesting to know what the strain is, because that can make the difference between having the disease or not having C. diff disease. And so when you're identifying organisms, really, if you really want to get down to strain level, we're going to have a little bit of a different approach than what we might have had in the past. So in the next slide, I show that this is possible, or we at least have evidence that this is possible. About a quarter of a century ago, Volker Gertler published this paper basically showing that for Clostridium difficile, and we'll use this as an example throughout, that you can tell the difference between different strains by simply looking at a combination of the 16S and 23S genes. Now, in the figure below the paper, there is a screenshot of part of the C. diff NAP11 genome, and these circles cover 16S, this small purple bar on the left, and the associated 23S genes. They usually come in a little gene cassette like that. And you can see on the leftmost circle that there's a little space between the 16S and 23S genes. And if you look in the one in the middle, if you look up just a little bit, you can see that there is a tRNA in that spacer region. And then on the third cassette on the right, you can see that once again, we're back to no tRNA in there. And so these are highly variable even inside one organism. And so what Volker did back then was do a PCR against the intervening sequence, that spacer region between the 23S and 16S gene. And he'd look at the restriction enzyme pattern. And then using that, he could tell the difference between the different strains. We can do that also with sequencing, but to an even higher degree of accuracy. And so in the next slide, I want to show what that actually looks like. So in the box right to the right of best match, there is a read that is mapped to the Z31 chromosome, and you can see it matches pretty well. So the long purple bar is the 16S gene, and then there's a little space, and then there's a start of another purple bar, which is the 23S gene. And you can see that our gray, that gray bar along the bottom of that box actually is the sequencing read, and it covers the full 16S gene and crosses through into the 23S gene and covers that spacer region in between. Now, if you go down to the part marked next best match, what you can see is that same read mapped to a slightly different C. diff genome. And the bracket shows that when you compare it to the read up top, or to the mapping up top, that there is some difference in this intervening spacer region. And then if you blow that up a little bit, follow the arrow over to the right, and the sequence down below shows you where the insertions, deletions, and mismatches are between these two different strains. And so what we have here is a practical example from sequencing a read that matches the Z31 C. diff strain is very different, or different enough from the strain 1071 that we can tell them apart. So I just wanted to show that this type of strain level identification is possible, but it really depends on the amplicon you choose. And so in this slide, we see in the center, you can see that familiar purple and gold with a V1, V2, V3. And then that's the 16S gene. And then there's the internally transcribed spacer in pink. And then to the right, it started with the 23S gene. So this shows you where our various amplicons that I'll be discussing are placed. You can see the V4 amplicon, which is, and the V1, V3 amplicon, which are 292 and 526 bases respectively. And these are typically sequenced using Illumina technology. Below is the about 1500 V1, V9 amplicon, the purple box. And then there is a darker blue box with the, what I'm going to call the EXT amplicon, which is 2,455 bases long, and it spans the full 16S gene, the ITS, and the 23S gene, a part of the 23S gene that you saw the example of the read up above. But the take-home message from this part is I really wanted to demonstrate, first off, that it's not unreasonable to assume that the ribosomal RNA gene sequence information can be used to differentiate between microbial strains. So with that said, we're going to get into sequencing that actually does this and the approach for that. So there are a number of challenges when you start a project like this. And really what you want to do, I think the best thing is to begin with the end in mind. What do you want out of the experiment? So what are the tasks that are going to have to get done or accomplished to get to a result that you're looking for? First of all, you got to get the DNA. Lysis and DNA preparation has been known for a long time in this field that the way you crack the cells is crucial to the result. Second, amplicon selection. We discussed a little bit of that just a minute ago. Depending on where the amplicon is and how much information it carries, you can get better or worse identification at the family, genus, species, or even strain level, depending on what you select. Primer design. Now, this is something I showed that there were conserved regions in the 16S gene. There are also conserved regions in the 23S gene. They're conserved, but not fully conserved. So certain families of organisms may have slight differences between them, and you'd want to cover all of the variability so that you can capture all of those organisms. And I'll discuss some unique ways that we have to do that. And then finally, analysis. You can have the greatest sequence data in the world, but if you're tossing all that up against a database that doesn't contain the organisms that you have or doesn't have the read length to distinguish them, you're only going to get a result that's as good as your database. So we'll take all of these in turn in the next few minutes. First, lysis and DNA preparation. And really the upshot is if you don't get the DNA out of the cells, you're not going to get any sequence data. And this is a harder problem than at first I imagined it might be when we took this on. You'd think, hey, this is solved. This is not solved. And the microbiome presents a unique opportunity and problem in this area, because if you're just lysing a vat of E. coli, who cares if your lysis is not getting every bug? You're going to get enough plasmid or DNA out. But in this case, really, since you're scanning a diverse population and some of the most interesting bugs are the hardest to crack. So if you're looking at gram-positive Firmicutes and you're looking at Clostridia, they can be really difficult to open. And so if you have a lysis and DNA recovery technique that doesn't open the tough bugs, sometimes these are the most interesting ones, and they'll be missing or greatly diminished in your final data set. So you want to have a lysis that will crack the bugs. And really, on the left, you have reality, where you have a very diverse array of organisms. And then on the right, typically what you see with what we've seen with most kits is that you're simply missing or greatly diminishing some of the more important organisms. Now, the hard part is that boring ones, they show up really easy. The more interesting ones can be a real problem. So we set out to address this challenge, and this is really where we made some of our first big gains. What I'm showing here is 5 different fecal samples. So these are actual real-life fecal samples that were prepared with our rapid prep versus one of the standards out there for this. And really, what we're looking at is the orange bar when we compare samples 1, 2, 3, 4, and 5, and the size of that orange bar relative to the rest of the microbes. And what you can see is that in the rapid prep compared to the standard prep, in almost every case, you see a lot more of the Firmicutes, the gram-positive ones, the ones with the tough cell walls coming out. So this was really the beginning of this, but of course you want to validate this and make sure that it's actually true. And one of the indicators early on is if you take a look at a sample like sample 4, you can see that there wasn't a big change. And so it's not uniform, and it depends on the proportion of bacteria in the sample that are difficult to lyse. So if you have a large proportion that are difficult to lyse, you'll get a sample that looks like number 5, where with the standard prep, you get a few of them. And then what that microbiome really looks like is that it is the majority difficult to lyse organisms. If they're relatively easy, all easy to lyse, you'll get a sample that looks like 4. And then typically what we see is something that looks like 1 or 2 or 3, where we're seeing more Firmicutes pop up with our rapid lysis method than with the standard methods. So to dig a little bit deeper in that, you'll think that, hey, if there are more of these Firmicutes appearing, do you get more Firmicute species with the improved lysis? And the answer to that is yes, kind of. So you get more of the really difficult ones. So the Clostridia I'm showing up top, those two, the Blautia and the Hadris, those two guys, what you see for most of the samples that we ran is that they're heavily underrepresented in the standard prep, and the representation goes way up with the Shoreline biome rapid prep. That's especially true with the Hadris and microbes of that type. But there are a number of them, like Streptococcus, that are equally represented regardless of the method, and that's exactly what you'd expect. Easily lysed organisms should appear in both lysis techniques, and they do. Now, the next question is, how do we know we're getting everything? And that's a real problem, because how do you ever know you're getting everything? So the way we decided to tackle this was to put together a completely independent lysis technique. The one I've been talking about is chemical. The one I'm about to talk about is physical. And we figured that if we have a chemical technique on one hand and a physical technique on the other hand, and they can give you the same results, chances are you're looking at most of the microbes. And the catch with the physical is that we wanted to be able to recover spores. Why spores? Spores are really hard to crack open without destroying the DNA. Many of the methods that you can use to completely destroy regular cells don't even touch spores. And that was the case in this case. And this is where our collaboration with Dr. Setlow's lab came in. He's been cracking spores for years. And so essentially we came up with a mechanical method for treating cells that would crack the spores, but not harm the DNA. And it's something that it's a unique little machine that has quite a unique action to it. It's we've applied for a patent on, but essentially what we can do is in just a few seconds, we can crack open spores. And if we get spores, then we get every other vegetative cell, or so we think. But we can also keep the DNA intact, which is the key, because a lot of bead beating methods or mechanical methods, when you do enough damage to the spores to get the DNA out, you've destroyed the DNA of the other cells that are in there. So we know from experiments we've done that we can quickly release the DNA and it's intact. And in the gray graph on the left, the Essentially what you're looking at on the left is, left axis is terbium fluorescence, which is a surrogate for DPA, which is a chemical that is packed around the DNA inside the spore head. And so we know we're tracking spores when DPA is released. And then we can look for the DNA, because we can release the DPA, but if the DNA is shredded beyond all repair, then we're in trouble. But what we can find is that even with extended treatments in this method, we get high quality DNA that is trapped in the wells. It's high molecular weight when you run it on a gel. And we crack the spores in just a few seconds. So we know we have a chemical method that does pretty well. And how does it compare to this mechanical method, which is suitable for opening spores? That's what you see on the right-hand side is the same samples done with the rapid prep, which we just went through before, a chemical method, and then the bead beating prep, which I just talked about here, which cracks spores. And what you can see in sample after sample is that the results are pretty much the same. And that's looking at the phylum level. So that's a pretty 10,000-foot view. But if you zoom in and take a look at the species strain level, you can see that once again, you're picking up the same organisms at about the same ratios, whether you're using the mechanical method or the chemical method. And so this says to us, we've got two completely independent methods of cracking open bugs. They give us a whole lot of Firmicutes and Clostridia, which are the tougher ones. We know one of these will give us DNA from spores, and they both produce the same results from the sample. So we're pretty confident that we've got a pretty valid way of cracking spores, cracking cells, and getting the DNA out. So the conclusions on this is that your results start with cracking open the cells, and if you don't lyse it, you're not going to sequence it. So we came up with a few methods that enable us to get the same answer regardless of how we crack the cells and recover the DNA. And if you're doing an experiment of this type, it is extraordinarily useful to have two independent methods of recovering DNA that yield the same answer. So that's really where I want to move on from lysis and move to the second challenge, which is Amplicon selection, read length versus read quantity. So essentially, the longer reads will give you more information, but they're going to cost you more money. So what is that balance, and how do you want to do that? So going back to this slide where I discussed the common Amplicons, the 292 V4, very common for Amplicons being sequenced using Illumina. And for V1, V3, it's a little bit longer, a little bit less common, but very powerful for getting more information out. And that's around 525 bases or so. Those are both Illumina. If you look down below, we have the V1, V9 in the purple box, which covers the entire 16S gene. More information than you get typically with either the shorter Amplicons, but still difficult to tell strain level organisms apart. And then down below that, we have the EXT Amplicon, the dark blue bar with the white lettering, and that's around 2,500 bases. And those longer Amplicons are typically sequenced using the PacBio platform. And that's some of the data that I'll be showing you. And that's where some of the more interesting results are coming. So I want to focus on that for a little bit. So what does it look like? Just to remind you, here's another example of a C. diff where we map a read that came from a strain of C. diff to a reference that's from a different strain. And you can see in this one, once again, that gray bar below the 16S purple bar. And that yields a map that actually contains a gap, in this case, in a base change. It's sufficient to tell the difference between the two strains. So we know, once again, here's another example of the ability to get strain level information about Clostridium difficile. And we're using this as an example, and for the reasons for that, I'll go into in a minute. But the trade-off. So like I mentioned, if you're going to sequence using Illumina, you're going to get a whole lot of reads for your money. And you can cram in 384 samples or more in a run. And you're going to get good sequence data for those V4 or V1, V3 or other short Amplicons. If you really want an experiment that's going to nail things down at the strain level, you're going to need longer amplicons, and typically PacBio is the way to do that. The sequel instrument that is some of the data that I'll be talking about with you today, you can put in about 100 samples on that and get fair levels, pretty good levels of information out. So that's what I'll be talking about mostly down below. The next challenge, primer design. So structure and function are related. And this is just something I want to take on for a minute, because remember in the beginning when I showed the 16S ribosomal RNA sequence, linear, and the 2D structure, and then the 3D structure. When you choose a primer that's in a conserved part of this ribosomal RNA gene, what you're essentially doing is choosing something that is an important structure, because any changes to that sequence will change the structure. Any change to that structure will make the ribosomal RNA non-functional, and therefore the organism won't survive. So in the upper right-hand corner, there's that little hairpin loop that I've put together, and that is a representation of the primer 27 forward site, which is in the V1 region, and it forms this hairpin loop structure that you can see. Now, the purple bases are completely conserved. If you take a look at the SILVA database and you take a look at all the available 27F sequences, you'll see that these are invariant across all the organisms in that database. Likewise with the Riken genome database. Those don't move around very much, but the bases in black do change, and the changes that are in the Riken database and in SILVA database are indicated by the degenerate letters for the nucleotides that are shown there. And you can see that it forms this little structure. And so the canonical sequence for the 27 forward primer is shown. Right below it is the in little red letters is shown the same sequence with the degenerate primers thrown in there. And if you were to take all the potential degenerate primers that you can make with those base substitutions, you'd get 384 different primers. If you're designing a PCR, this is a pretty inefficient thing. You don't want to have 384 primers in there. So we took a look and said, hey, how many of those 384 are important? Like, for instance, how many of them actually exist in which organisms, and then maybe we could sort things around. It turns out that if you start really looking hard at this, there's only 17 allowed variants within all of that 384 base space. So we were able to cover all organisms in the Riken database and all organisms that contain this primer sequence in the SILVA database with only 17 different primers. And so this suddenly becomes tractable. You can design a PCR that's got 17 degenerate primers in there. And why would we want to do this? Well, we know that there is variation from organism to organism, and that even a 2 base difference in the primer sequence can eliminate the representation of certain microbes. And in addition, the search revealed that there isn't any published primer pool that covers all the known organisms that have been sequenced that are part of these databases. So what we did was we went back and designed primer sets that match all the variants for the relevant primer sequences that I've been talking about. So down below in the lower right, you can see where the 2D structure sort of melds into a 3D-ish structure, base pairing between different parts of the 16S gene, and why those changes may be linked in the, to other changes inside the ribosomal RNA gene. In addition to designing for the known variants, because we know we need to maintain this structure and these base pairs within the 16S gene, we can also potentially design for variants that we haven't found yet. So you can look at this and say, what would the base changes that would be allowed that would still maintain these structural relationships and incorporate those as well. So it's a very interesting set of findings that we're able to take advantage of. So that's primer design. Now I want to talk a little bit about barcode scheme. What are the barcodes? Well, the barcodes are little DNA snippets that are attached to each sample so that we can sequence them all together. That makes things cost effective. But then later on, we can read those barcodes and then resort all of those reads into their original samples. And so there's a couple of different methods that are out there. One is combinatorial, and this newly published one by McConnell and all are this unique dual matched. And really the difference is that on the left, that little figure down below, the little colorful figure says combinatorial. You're using across each column and row, you're using a single barcode. So if you look down in the first column, you can see that the top half of the circle is black. And if you look at the top row, you can see the bottom half of each of the circles is white. And it's the combination of the two that tells you which sample the read originally came from. So those are attached during sample prep, and then they're mixed together and sequenced together, and then you read the sequence to sort them later. With the unique dual matched, it's a little bit different in that every sample has two unique barcodes, one on the front and one on the back. And so if you read it to assign a sample to a well, you have to get both barcodes correct. And so the chances of misassigning a read are much lower when you have two cross-check barcodes as opposed to just one. So the problem is that the combinatorial is simple and cheap to do, and the unique dual matched is much more expensive and harder. So depending on the degree of accuracy you need for your experiment, you'd want to take on one or the other. So that is barcoding, and then I want to move into analysis. So analysis, even if you have the best data, it's not going to necessarily get you this kind of result, where those who are familiar with Mother will recognize that this is kind of the output that you get. Across the top, you can see that there's the taxon and sample 1 and sample 2 in this case. And then for each taxon, it tells you what the microbe is. And so in this case, we've got a high degree of we've got extra long reads here, so we can map down to the strain level. This is a database that we put together at Shoreline. We're calling it Athena, and it contains these linked 16S, 23S gene cassettes. So when we map long reads, we can get the kind of accuracy that we would want to have at the strain level. And so now I'll get into, to continue this thought, I'll get into why are we using Clostridium difficile as an example. Because there's a whole lot of sequencing that's been done, and it's relatively simple to validate that we're getting the correct answer. And so if you take a look, the taxonomy of Clostridium difficile is shown in red across the different levels of taxa, from family down to genus species. And inside the Clostridioides difficile, there's 912 different genomes that have been sequenced to one degree or another. And around the outside of that taxonomic tree, that circle down there, you can see I've put in black where the fully sequenced genomes are on the taxonomic tree. And so it gives us a lot of known diversity that's really heavily sequenced, so we can tell, are we getting the right answer or the wrong answer, because there's a lot of data here. So when we take a sample that contains C. difficile, we should be able to sort out which strains of C. diff are in that sample. And that's what I'm going to show you in the next slide. So your database, for each of these three different databases we map to, the ribosomal protein database, Silva, and Athena, which is the Shoreline Biome database, we essentially took the same data sets with the C. difficile reads, and we mapped them against these databases to see what it would produce. And so the shorter reads that are in the RDP and Silva databases, really, they will get us down to Clostridium. So it's some kind of Clostridium. And that's great. I mean, if that's what you need out of the experiment, that's fantastic. In this case, though, what we wanted to do was get down to the strain level. And why you'd want to do that is evidenced by this particular sample, because number one, there's 5 different strains of C. diff that this individual is co-infected with. They were at it was at a very low level. In fact, this was a sample that came back as negative when in fact we're able to detect a C. diff in it as positive. And what we're able to show is that there is the presence of this C. diff Z31 in there, which is a non-toxigenic strain. That is, it doesn't have the disease-causing toxins in it, but it's also co-evident with a number of strains that are just plain bad news. So this kind of thing tells you that even at the genus species level, you have C. diff. There are different strains of C. diff that may have different effects. So if you're inhabited with C. diff Z31, it doesn't have the toxin, and you're probably not going to get the disease. The other ones, yes, you will. So having the database that has these longer gene cassettes in it enables us to really get down to the strain level of identification, which is great. And so what we want to do, though, is this was a really well sequenced example. There's a lot of C. difficile genomes out there. What about other genomes? Can we, organisms, can we generalize to other organisms? And I think the answer to that is yes, because if you take a look at this, we knew that the organism was supposed to, could be there, even though it tested negative. The person had something going on. And so we detected this at low levels at 1.4%, and we're able to get 5 strains of specific information out. And anything that wasn't assigned as a strain was also assigned as C. diff, the remaining few reads. So what we know we can do is, because of these long amplicons that we've got that cover this and internally transcribed spacer into the 23S gene, we can actually even take unknowns and categorize them as well, because you might not know what it is, but you've got enough sequence information to know that it's not anything that's been sequenced before. And so if that's an important organism to you, you can follow it, track it, bin it, and potentially even sequence it if it's interesting enough. So what do you get when you put it all together? On the right-hand side, what I'm showing is a short amplicon with standard lysis techniques. And what you can see is that for some of these samples, the first sample on the left and the third from the right, it doesn't look very interesting. But if you combine the optimized lysis and you've got a long amplicon and you've got a good database, what you can do is you can take a look at sample number 1 on the left with the long amplicon and the optimal lysis, and you can see that that's far more interesting than it first appeared. There's actually very little of that sort of gold boring Bacteroides in there, and it's mostly made up of other organisms that would be extraordinarily interesting to track down. So once again, beginning with the end in mind, the experiment looks very different at the end, depending on the choices that you made at the beginning going in. And so the analysis, the effect of the analysis, or the outcome of the analysis, is going to be very different. So that's really putting it all together. And just to come back to what the challenges were and to reiterate, it's important for lysis of DNA preparation. If you don't get the DNA, it's simply not going to show up. Your amplicon selection is important, depending on how far you want to get down there. Your primer design, how are you going to cover all those microbes, right? Those conserved sequences aren't necessarily fully conserved, so you've got to put some degeneracy in there. And then for your analysis in this case can be as good as the database that you're using. So it's important to consider that when you are putting your experiment together. How are you actually going to take a look at these reads when you're done? And then what I want to do is just talk about the reasons we are taking this on at Shoreline is because we wanted to put together a process and a kit that would cover all of these issues and put it in something that you could just put your sample in, have all of this taken care of, choose your amplicon, and have it come out. And if you are interested in this and finding out a little bit more, you can shoot me an email. There's an email associated with the talk today, but also you can feel free to send an email to labroots@shorelinebiome.com to me, Mark, with any comments and questions. And with that, I'll be glad to take questions.
Thank you, Dr. Driscoll, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many questions as we have time for. Let's get started. Our first question is, you spend a lot of time on lysis methods. I thought there were microbiome specific kits and protocols available that already solve this problem.
Yeah, that's a great point. We thought the exact same thing until we really got into this. I was really convinced that the lysis problem was solved. Some of the very first experiments we ran, we saw that this actually isn't the case, and that we were missing a lot of the really important Firmicutes that we really wanted to find out information about. So we tried a wide variety of different kits. Those results are currently being collated and published, or going to be submitted for publication very soon. And so we should see some more information out there about this. There's a number of publications that I didn't directly reference, but are in the presentation that talk a little bit about the lysis problem, or the missing Firmicutes problem. So it's something that's known. I think people have been working around it, and we tried our best to actually solve it.
Great, thank you. Another question we have is, to cover the known microbial diversity, you discussed using degenerate primer PCR in your 16S microbiome assays. However, the ratios of degenerate primers can introduce a new kind of bias based on relative degenerate primer concentrations. Do you address this, and if so, how?
Yeah, that's something that we struggled with a bit, because as you introduce degenerate primers, as I was talking about, you can get up to, it looks like you've got to add, in some cases, 384 primers to cover the degeneracy. So when we really dug into this, I addressed a little bit in the talk about how we can cover all known diversity with far less primers. In fact, I think the number was 17 for the V1. But even that, even having as few as 17 primers in the mix can result in primer-to-primer bias. So what we've done is we've come up with a unique PCR approach that addresses this, where in the beginning, the PCR amplicons are developed by specific primers, but then as the reaction proceeds, sort of a non-specific PCR reaction takes over and proceeds to amplify everything to the end of the reaction while maintaining the ratios in the beginning. So in that way, we're able to avoid most of the primer diversity bias that comes from having a lot of different primers in there. It's something we're going to be publishing on very soon, so stay tuned.
All right, and we have time for one more question. And this one says, it seems like a lot of the field is moving towards shotgun microbiome sequencing, but your presentation is focused on 16S. Can you comment on the relative merits of 16S versus shotgun approaches? Why keep using 16S?
Yeah, that's something that's known. There's a trade-off with 16S, just simply because you need a lot less sequencing. So with the shotgun microbiome sequencing, there's hundreds to thousands of times more sequencing information you need to get the same thing and the same result. So you can do a lot fewer samples that way, which is kind of an impediment. The other issue was really, are we covering everything? And that's something that I think we're able to address more or less with our primer diversity, where we're covering everything that's been sequenced, and we're potentially able to cover things that haven't yet. So there's that as to why you'd want to continue doing this inexpensive nature and improvements to the diversity output. Really, the issue is also with the database. As I discussed at the end, the database that you're comparing to or mapping to really allows you to identify what organisms are in there. And some of the experiments that we've done with the shotgun, even the best databases for that we know we put in only 10 organisms and a few hundred come out. So we know there's a huge disconnect in there as to how do you assign which organisms are present. And so that's a problem that in the whole genome sequencing shotgun microbiome is going to have to be better addressed, I think. So there's a number of reasons, but really it's, once again, getting back to, what do you want out of the experiment? If you want to identify the microbes, sort of a fast, cheap way to do it is 16S still.
I would like to once again thank Dr. Mark Driscoll for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues and who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.